Welcome to G Fun Facts Online. Today, we're diving deep into the brain, exploring a condition that touches millions, Alzheimer's disease. It's a relentless challenge, stealing memories and cognitive function. But science is fighting back, and the battlefield is incredibly complex, involving our genes, our brain's own immune system, and even the cells designed to protect our neurons. We're going to uncover the fascinating link between a specific gene called apolipoprotein E, or APOE, Alzheimer's, and the brain's resident immune cells, the microglia. This is cutting-edge stuff, so let's get into it. First up, what exactly is this APOE gene? Well, it's a crucial player in our overall health, primarily known for its role in transporting lipids, like cholesterol, around our body and, importantly, within our brain. In the central nervous system, APOE is mainly produced by cells called astrocytes and, you guessed it, microglia. This protein is vital for keeping our synapses flexible, repairing neurons, and maintaining overall brain balance. But here's the twist. The APOE gene isn't one size fits all. It's polymorphic, meaning it comes in different forms, or alleles. The three common human versions are APOE2, APOE3, and APOE4. They differ by tiny changes in their structure, just single amino acids, but these small differences have huge implications for Alzheimer's risk. Now let's focus on APOE4. This particular version has emerged as the most significant genetic risk factor for late-onset Alzheimer's, which is the most common form of the disease. If you carry one copy of the APOE4 allele, your risk of developing Alzheimer's is substantially increased. If you have two copies, the risk is even higher, and the disease might even show up earlier in life. On the flip side, the APOE2 allele is actually considered protective. It's associated with a lower risk of Alzheimer's and even linked to longevity. APOE3 is the most common version and is generally seen as having a neutral effect on risk. So how do these APOE variants actually change our risk? That's where microglia step onto the stage. Microglia are the brain's primary immune cells. Think of them as vigilant security guards, constantly patrolling their environment for any signs of trouble, injury, infection, or the buildup of harmful proteins. In a healthy brain, microglia are heroes. They clear out cellular debris, help prune unnecessary connections between neurons during development, and launch an inflammatory response when pathogens invade. But in the context of Alzheimer's, and especially when influenced by different APOE isoforms, microglial behavior can become a double-edged sword. One of the telltale signs of Alzheimer's is the accumulation of amyloid-beta plaques in the brain. These are clumps of protein that disrupt brain function. Microglia are naturally equipped to recognize and clear away amyloid-beta. However, how well they do this job seems to heavily depend on their APOE genotype. Studies suggest that microglia expressing APOE4 are less efficient at gobbling up and degrading amyloid beta compared to microglia with APOE2 or APOE3. This inefficiency can lead to more amyloid beta plaques building up, essentially pouring fuel on the Alzheimer's fire. But that's not all. The interaction between APOE4 and microglia also promotes a pro-inflammatory state within the brain. Microglia can switch between different activation states. Broadly, you can think of them as either pro-inflammatory, like an attack mode, or anti-inflammatory and pro-resolution, like a healing mode. In the presence of APOE4, microglia tend to get stuck in that aggressive pro-inflammatory state. These revved-up microglia release a barrage of inflammatory molecules, things like cytokines and reactive oxygen species. While these are meant to combat threats, a constant onslaught creates a chronically inflamed environment that's toxic to neurons. This sustained neuroinflammation contributes to neuronal damage, faulty synapses, and ultimately, cognitive decline. Recent research has added another layer to this. APOE4 messes with lipid metabolism inside microglia. Microglia with APOE4 tend to accumulate more lipid droplets, essentially stores of fats, than those with APOE3. This isn't just a passive buildup. It actively drives microglial dysfunction. It can trigger inflammatory signals, increase oxidative stress, and further hamper the cell's ability to clear amyloid-beta and other waste. Some studies even suggest these lipid-loaded, dysfunctional APOE4 microglia can release substances that are harmful to neurons. It's a complex web because APOE itself is all about lipid transport, and its different versions handle this with varying efficiency. Those tiny structural differences in the APOE4 protein are thought to be the root cause of its damaging effects on lipid handling and its interactions with receptors on microglia. And the story gets even more nuanced. Scientists are using advanced techniques like single-cell RNA sequencing, and they're discovering an incredible diversity in microglial states, moving way beyond the simple pro-inflammatory/anti-inflammatory labels. They're identifying specific microglial subtypes associated with disease progression, sometimes called disease-associated microglia, or DAMs. APOE signaling is crucial in guiding microglia from their normal homeostatic state to these DAM states. For instance, another important Alzheimer's risk gene, TREM2, which is highly expressed in microglia, interacts with APOE. Activating TREM2 can trigger an APOE-dependent pathway that influences how microglia respond to amyloid-beta and neurodegeneration. If this TREM2 APOE pathway isn't working right, it can impair microglial activation and their ability to clean up and form protective barriers around amyloid plaques. When we talk about the evolving brain here, it's not just about how the disease progresses in a person, but also about our own scientific understanding, which is constantly evolving. Researchers are continually peeling back new layers of complexity. For example, recent studies using human-derived microglia, sometimes grown from stem cells and even transplanted into mouse models, are giving us much more human-relevant insights. These models have shown that APOE4 microglia produce more inflammatory cytokines and are less able to switch into protective modes compared to APOE2 microglia. Conversely, APOE2 microglia seem to have boosted protective functions like better proliferation and migration. So what does all this mean for treatment? Well, this deep understanding of the APOE microglia axis is opening up exciting new avenues for therapeutic intervention. Several strategies are being explored. One approach is to modulate APOE4 function, trying to convert APOE4 into a less harmful, more APOE3-like or APOE2-like form using small molecules, or even reducing APOE4 levels with gene editing technologies. Another is targeting microglial activation, developing drugs to calm down those harmful pro-inflammatory responses or nudge microglia towards more neuroprotective states. Given the role of messed up lipid metabolism in APOE4 microglia, interventions to correct these metabolic imbalances are also on the table. Drugs that interfere with lipid droplet formation have shown promise in early studies. And finally, some strategies aim to boost the microglia's ability to clear amyloid plaques and other toxins, perhaps by targeting receptors like TREM2 or another receptor called LILRB4, which can be inactivated by APOE. Antibody treatments blocking the APOE-LILRB4 interaction have shown promise in mouse models, allowing microglia to clear amyloid more effectively. Unraveling the intricate roles of APOE and microglia in Alzheimer's is a journey that's far from over. But every new discovery brings us a step closer to understanding the fundamental breakdowns in the brain. Focusing on how our unique genetic makeup, specifically our APOE genotype, shapes the behavior of our brain's immune cells, offers a personalized way to look at disease risk and progression. This evolving knowledge holds immense promise for developing targeted therapies that could one day slow, halt, or even reverse the devastating course of Alzheimer's disease, bringing hope to millions. The complex dance between APOE, microglia, and the evolving landscape of the brain remains a critical frontier in neuroscience research. Thanks for tuning in to G Fun Facts Online. We'll catch you next time with more fascinating science.